labia minora. These flaps, one on either side, they kind of enclose that opening there that includes the vaginal and urethral orifice. That's the vestibule, just that kind of whole open area between the labia minora on either side. They're just covered with mucous membrane. Labia minora is not pigmented, no hair follicles. And if you look at the labia minora anteriorly, so here we're moving up, forms a little hood. That's the prefuse in the female. It's just a little fold of tissue covering that little bit of erectile tissue there, which is the clitoris, which is made of similar erectile tissue as the corpus spongiosum in the male. There you can see cross section. That's what most of our models look like. If you move out from the labia minora, there's the two labia majora. Those are just covered in integument, pigmented skin, hair follicles, and so forth. And really the last part to mention, here's a pubic symphysis. I don't think I have it on your lab sheet, but that pad of fat right in front of the pubic symphysis, that's considered part of the female external genitalia. It's the mons pubis. Just that area of fat right there. And that's really all we need for the female external genitalia. What we'll look at next, though, I want to look at their development a little bit because it's kind of cool. That is, you're looking at male, females, genitalia, they look quite different. But if you look back developmentally, you go early in embryo, let's switch back to early embryo, you can't tell the difference between different ones that I like better color. You can't tell the difference between the male and female in early embryo. It's about a 5-week embryo at the top there. 5-week embryo. They refer to this as the indifference stage. So it doesn't matter whether you're male or female, everything looks the same at this point. So if you get an ultrasound 5 weeks, they're going to tell you it's a girl or boy. You can't tell. Now it's kind of cool. What determines whether you're a male or a female in a human? What's the difference? Do you just randomly develop a male or a female, or it's determined by? Alright, it is a genetic difference in males and birds, but it's surprising how little a genetic difference does. You have male, there's different sex chromosomes in both males and birds. Males have an XY, that last pair of chromosomes. Females have 2 X's. If you look at these chromosomes, of course, chromosomes have genes on them. In the females, the 2 X chromosomes are basically a homologous pair, right? They have similar genes in the same location. In the males, while we treat this as a homologous pair in meiosis, it's not really. The X and the Y chromosome don't have the same genes on them. In fact, if you think of the Y chromosome, are there any genes on the Y chromosome that are essential for, I guess we call normal physiology, normal functioning, your body systems and whatnot? There can't be, or most of you would be dead because you don't have one. So there can't be anything all that critical on the Y chromosome, right? Females don't have it. The X chromosome, however, you must have it. You can have genetic anomalies where someone gets just an X chromosome, nothing else. They will still function. But if you get just a Y chromosome, no X's, the embryo won't develop very far. You're not going to make it. So the X chromosome does have some essential genes. The Y chromosome, of course, they can't be essential. Half the population doesn't have them. So what's on there? Well, honestly, they don't know. There's a lot I'll call it joke. That's not what the molecular biology would call it. But there's a lot of stuff on there. It may be sequences of DNA we don't use anymore. Maybe they're regulatory. I'm not really sure. But there's really only a few genes in it. And of course, they're the ones that will determine whether you develop a male or a female. And one of them we call it the HY antigen. Your book will use that. That's used in general, mammals in general. Name for it, humans, I just can't remember. There's a gene that produces this protein, HY antigen. That's the only difference between the male and female. That Y chromosome expresses one gene right about at this point in development. That gene's expressed, you should develop as a male. If it isn't expressed because you don't have it, you develop as a female. Now the way it works is kind of cool. All right, let's say we go to this 5 to 6 week stage here. I'm going to zoom in a wee bit. All right, they're kind of very diagrammatically showing you some of the reproductive structures. These kind of in purple here, Mullerian duct or paramesonephric ducts. That purple layer, that's going to develop into all the female structure, uterus, uterine tubes, and so forth. The ones kind of in, I guess, green there, those are the mesonephric ducts. Those tend to develop into male structures, vas deferens, several vesicles, and so forth. And then the gonads, there's a whole strip of tissue here and here. These are referred to as indifferent gonads. At this point in development, 5 to 6 weeks, you got all the tissues to make an ovary or testes. They're both there. Now what happens, shortly after this stage, let's say 6 to 7, next week, the gene on the Y chromosome will become active and it will start producing its gene product, this HY antigen. That acts as a signal. What it does is it causes the indifferent gonad portions of it to develop into a testes, the part that's going to be ovaries degenerates. That's it for the genetics. Once you have testes, of course, what do testes secrete? So keep in mind, this is only about 7 weeks of development or so. The testes will start cranking out testosterone. That's the key. If you have this secretion of prenatal testosterone, you will start to develop into a male. Zoom out a little bit. So what they're showing you here, this is after week 6, so 7, 8, and then at birth. If you're a male, you have that Y chromosome, it's going to produce this HY antigen, causes the testes to develop. Once the testes develop, they start producing testosterone. Testosterone is actually the signal. If testosterone is secreted, all of these parts, these Mullerian ducts that would have been female reproductive structures, what they're trying to indicate with all those breaks there, that part degenerates, it doesn't develop any further. The mesonephric ducts, that will be the male system, they're going to grow into epididymis, vas deferens, seminal vesicles, and so forth. If this were a genetic female, there's going to be no HY antigen. Sort of by default, the parts that are going to be the testes degenerate, the ovaries are retained. If you have ovaries, you're not going to produce testosterone. So if there's no testosterone, it's just the reverse. Those Mullerian ducts tend to be retained and reinforced, the mesonephric ducts degenerate, and so you'll end up with the uterus, uterine tubes, and so forth. Externally, it's just in the part. Almost the same thing happens. Next slide. This is showing the external genitalia. Basically, same age, is 6 weeks on the indifferent stage. So this is what everybody looks like, regardless of sex, early development, about 6 weeks. But again, after the indifferent stage, if you're a male, you'll have that HY antigen, testes will form, you'll start producing testosterone. Now what they're showing you here with all the color coding in this diagram, they're showing you homologous structures, males and females. What a homologous structure is, it's basically a structure that has the same embryonic origin, but it has different functions, as an adult, I would say. An example would be, a human hand and a bat's wing, those are considered homologous structures. If you look at, all the bones in your hand, the phalanges, the carpals, and so forth, you can compare them to a bat wing. They have the same bones in the same position. Their fingers are just elongated, and there's, membrane between them, but they're the same, embryologically, developmentally. Bat wing, human hand, same stuff. That's a homologous structure. So you can trace it back, it's got the same origin. Even though we use our hands for grabbing things and bats use them for flying, so they're functionally different in the adult, they start off as the same structure. Same is true here with these reproductive parts. So I'll name a few, and then we'll just follow them, see what they end up developing in male or female. Little kind of yellowish bump there, that's genital tubercle. There's two pictures over here. There's my genital tubercle. Tubercle's just a bump. There's a little bump there. Then, on either side, I've been bluish on this diagram. Those are labioscrotal folds. Genital tubercle, labioscrotal folds. If you move medially a little bit, there's a couple of these purple folds of tissue. Those are the urogenital folds. All that's different colors, so I can red again. And there's a groove in the middle, kind of a hollow spot. That is the urethral groove. It's just an opening. And that's what we'd all look like at about 6 weeks. Then if you're a male, and that testosterone is secreted a little bit later. If you follow these parts, of course, look at the genital tubercle. When you look at, mature, someone is born, postpartum, the genital tubercle is a gland's penis. So the head of the penis is the genital tubercle. The urogenital folds here, what they do is they elongate and they fuse together, except for the very tip, which is the opening of the penile urethra. So the urogenital folds fuse together, elongate, and that's basically going to form the shaft of the penis. And then, of course, the labioscrotal swellings, kind of self-implied in the name, that's where the testes will drop into and they form the scrotum. Now, if you're female, you start off with the same part, but there's no testosterone. So if you follow these same pieces, the genital tubercle ends up being the clitoris in the female. So the homologous structure, the clitoris in the female, starts off as the same structure embryologically as the gland's penis in the male. It's that genital tubercle. The labioscrotal folds and the testes, or the scrotum in the male, those are the labia majora in the female. So those are homologous structures. And the urethral folds don't seal up, they stay separate. And those end up developing the labia minora in the female. So labia minora and basically the shaft of the penis are homologous structures. So that's all just due to hormones, really. I mean, yes, there's a genetic difference. That's what sets us in motion. But the genetics doesn't make all the parts develop. It's the presence or absence of testosterone. Which means, let's see, there's a weird thing. It doesn't happen in humans, but it happens in cattle. I'm not a rancher, so you correct me if I'm wrong, but I believe they call them pre-market twins. What happens in cattle will illustrate this pretty well. The cow. It's a lot like my pregnant lady, but it's a cow. And cattle will often have twins. There's a placenta, there's a placenta. So I'll put a little cap in here. And I'll put another little cap in here. Let's say for this calf's a male, this one's a female. All mammals follow the same pattern. Yeah, the timing's off, and I don't know what it is in cattle because their gestation period is different. But these things will start off, within the different states. The males have an X and Y, females have an XX. But unlike humans, because this wouldn't happen in humans, the placenta in cattle seems to be a little leakier than it is in some other mammals. So when this male reaches kind of this stage right here, of course the testes develop, he's going to start secreting testosterone, which is going to affect his development. He'll vascularize all his parts. But some of that testosterone can leak across the placenta into the mother's blood, and it will move across the placenta into this female calf's blood, and it will affect her development. And sometimes, in extreme cases, what I've heard when these things are born, you can look at them, and it looks like two male calves. They look the same. One of them is a genetic female. Typically, they're both sterile, and they have big breakfast orders than normal, but they're remarkable cattle. So it's all hormonal that causes these structures to develop. The genetics just kind of initiates it. I guess it'd be possible if you had a male, let's say, that had a mutation that had no receptors for testosterone. Guess what? When they're born into a female, they have all the genes on the genitalia. They may still have testes, but all the parts with a pure female. But testosterone, yes, it's responsible for male secondary sex characteristics, but it's also responsible for developing the primary male sex characteristics, causing all the gender to form. All right, and then the last thing we have to do with the reproductive system, endocrine control. Hormones control the reproductive system. I'll do the male first, because it takes like a minute, and then we're done. Female will take a little bit more explanation, and it's way better for short answer questions. Let's see. For the male. Down here on the x-axis, I'll put age. 0 is birth, so -1, you're not there at all, but so this is development back here. And then we'll need to keep track of FSH, LH, and testosterone. I can do both different colors. There. All right, really, in the male, all right, somewhere right around here, there's nothing there. Nothing, 5, 10, somewhere in here, you know, puberty kicks in, and then we just get something like this. All right, endocrine control of the male is pretty simple. All right, basically the only thing you have to sort of watch out for here, this represents testosterone levels, plasma testosterone levels. Notice it goes up real high. That's before birth. What's that recording? Recording that prenatal surge of testosterone that makes everything develop male. But then, you know, before birth it drops down and it stays low, nothing's happening. Till puberty hits and FSH, LH go up, testosterone goes up, and they basically stay up and they remain up. You know, I guess maybe or daily cycle a little bit, but it's pretty much elevated, stays there. And then of course physiological senescence kicks in and everything falls apart, so we kind of ignore that bit. But in males, it's pretty simple. You know, you hit, you know, sexual maturity, everything goes up, and everything stays up. It's all the same. So there's nothing going on from, you know, puberty until, you know, you start to fall apart and dying out at this end. So that's it for the male, piece of cake. All you gotta remember is that prenatal surge, that's the only kind of weird part. Now the females, way better for testing. This will take a minute. Let me put a diagram up, something like this one. You'll have one like it in this book. I can't remember exactly this one. All right, kind of looking at more or less the same hormones in the female. We'll look at plasma levels of FSH, LH, getting chromatotropics from that anterior pituitary. But then we have two female sex hormones, progesterone, estrogen, and we have a fifth hormone, HCG. All right, so we're going to keep track of plasma levels of eventually all 5 of these hormones. I've also got kind of some different areas to look at. The way they'll often separate this off, because there's a lot going on, they kind of separate it into different parts at first, makes it a little easier to follow. Of course, these are happening concurrently, eventually we'll have to put them all together. What we're looking at in this top box, this is sometimes called the ovarian cycle. What you see the bottom half of this top box, this is one we've already looked at. Look at the left, one over to the left a little bit. That's a primordial follicle, primary, secondary, mature, ovulation, forming corpus luteum. It then generates corpus albicans. So these are those stages we looked at in the ovary already. Notice there's a time scale at the bottom. Basically, the female reproductive system is on approximately a 28-day cycle. So you could I suppose, arbitrarily start anywhere, but they're calling this day 1, and it just loops around. Day 1's back here again, so it just keeps going around. So the female reproductive system cycles after sexual maturity at about a 28-day cycle. So again, just like the male, nothing really for puberty. Then all levels start going up, but unlike a male, they don't stay at a set point. They go up, they go down, they go up, they go down. So we're looking at changes in the ovary over that 28-day period. And the top half of this box, these are just showing you relative levels of follicle-stimulating hormone and luteinizing hormone. Luteinizing hormone is in red, follicle-stimulating hormone is blue. And you'll notice those levels are not staying constant, they oscillate. Bottom box, kind of showing you the other half. Down here, this is showing you the endometrial layer of the uterus. Remember, that's the part that will respond to estrogen and progesterone levels. You know, it's really thin here, that's that functional layer. It gets really thick, then it sloughs off, gets thin again. So that's, again, the same 28-day cycle. You're just looking at the endometrium of the uterus. They sometimes call this a uterine cycle. The top half of this bottom box, that is plasma levels of estrogen in red and progesterone in green. So I'll go over them one at a time, just to kind of give you the idea. And eventually we just have to look at them both because they're occurring at the same time. It's just easier initially sometimes to separate. So if you look at this top box, again, this is looking at those follicles in the ovary. We can randomly start anywhere. Day 1 is like right here. and it wraps around to right here. It's all the same. Continues. A couple things to note. If you look at day one, how would you describe the plasma levels of FSH and LH at that point, right about here? Does it get any lower anywhere else? No. Day one, FSH, LH levels are the lowest they're going to get. Really low baseline levels. And if you look in the ovary, the follicles aren't being stimulated. They need FSH for stimulation. So you can think of that as like a primordial follicle. It's just sitting there, there's a bunch. So as you go from day one through the cycle, one of the first things you'll notice, look at the levels of FSH and LH. They go up. They go up. We already know what FSH does. Under the influence of FSH, that's where you can see the growth of those follicles. They go from primordial to primary to secondary. They get bigger and bigger and bigger. Now, something else you might note on this diagram, I think they showed it on purpose. Notice FSH levels go up, but then they drop back down a little bit. Not really a mistake. It's not a huge change, but kind of what's happening here. In a human, how many follicles do you want to ovulate at a time? Generally, how many eggs ovulate a human? You usually want one, no more than two. We're not like a cat, man. You try and cram 8 embryos in our uterus, it can't handle it. It's not, it won't do it very well. When you hear those people having 8 offspring at a time, that's probably some in vitro fertilization going on, because we don't normally ovulate more than 1, occasionally 2 eggs. That's what the uterus is designed to handle. But what happens is that FSH doesn't stimulate just one follicle to start to grow. There's actually a number of a handful that are all starting to go from primordial to primary to secondary. When you reach ovulation, you don't want all these ovulating. Again, the uterus of a human can't accommodate that many growing embryos. So what happens, if you look at all these follicles, just kind of think of those cat ovaries. You'll notice there are a bunch of them are different sizes. Some of them are a little more sensitive, they've grown a little faster. Others are still developing, but they're smaller. As the FSH levels drop, what happens, only the follicles that are the most mature, they produce a lot of receptors for FSH. The slightly less mature follicles, even though they're still growing, they don't have as many receptors for FSH. So as it's kind of like upregulation in a way. As FSH levels drop, only the follicles that are furthest along that have produced most of the receptors, they will still respond to those lower levels of FSH. Some of the smaller follicles that start developing, they'll just stop. They're no longer stimulated, they degenerate. That's for sure. You know, the drop there. But it's still stimulating the follicle. It's still growing. It's still growing. Now, right here, of course, there's something going on. Say about the midpoint of the cycle, call it close to day 14. You'll notice there's a huge spike in the release of FSH and LH. I mean, FSH goes up a little bit, LH through the roof. That spike in FSH and LH, again, I think we talked about this at some point in lecture lab, I don't remember which. But that surge in FSH and LH makes that follicle grow so rapidly, it ruptures. That's what causes ovulation. So about the midpoint of this cycle, the increase in the secretion of FSH and LH, that surge, causes follicle rupture. That's the cause of ovulation. Now the oocyte, the egg, that's going to make its way to the uterine tubes. It may be fertilized, it may not. We'll kind of look at both situations down the road. But for now, it's out of here, so it's making its way through the uterine tube, and it'll take a few days to do that. Now, what you're left with, after that follicle ruptures, of course, you got this exploding sort of crater in the ovary. This is where LH comes in. These still somewhat higher levels of LH will target the ruptured follicle, and they cause it to fill in with endocrine cells and form this fairly large structure called the corpus luteum. So under the influence of luteinizing hormone, LH, the ruptured follicle fills in with cells, and it becomes corpus luteum, yellow body. Now notice though, LH levels are still dropping. It's LH that's targeting that corpus luteum. After, say, about 10 days, LH levels have dropped low enough. That's not enough to support the corpus luteum, so it's no longer going to respond because there's no LH. That's when it degenerates and shuts down. So you only get about 10 days of the corpus luteum. If LH levels drop, then the corpus luteum loses its function. It regresses, or we call it involutes, and it doesn't disintegrate, but it shrivels up, and that corpus albicans is just the remaining scar tissue from when that follicle prevents the rupture. And if there is no pregnancy, that's what happens, and you just go right back and start it again, again, again. Now, we'll move to the second box. There are a couple things to note. The follicle. That's the source for most of the estrogen. If you notice, as the follicle gets bigger and bigger and bigger, look down here in this box as we go from day one up to about the midpoint. Notice that estrogen levels go up and up and up. That's because the follicle gets bigger, secreting more, bigger, secreting more. The green hump here, which is the progesterone, that is secreted primarily, most of it, by the corpus luteum. So you notice you don't have real high levels of progesterone until the corpus luteum forms. That's where you get the max. So these hormones are actually coming from the ovaries, which we've already mentioned. And now, of course, the bottom layer, the endometrium, as I already mentioned, it's sensitive to estrogen and progesterone. If estrogen and progesterone levels are low, so we look at day one here, and it wraps back around over here. That's day one. Estrogen and progesterone levels are at their baseline. They don't get any lower than that. And because of that, you can't support a thick endometrium. Those cells lyse, they get reabsorbed, they get sloughed off. That's kind of what they're showing you here. This functional layer is actually flowing off. So I kind of show it won't be looking here. That's going to be the menstrual flow. We just continue with it on here. So right at this point, it's actually about day 3, menstruation is finished. The endometrium is at its thinnest. It's basically the functional layer, a few scattered, or sorry, the basal layer, a few scattered functional cells. So now as we go, after menstruation, from about day 3 onward towards the midpoint, of course that follicle is growing and estrogen levels are going up and up. So the estrogen, as we've already mentioned, targets a functional layer of the endometrium and it causes those cells just to proliferate. And that's all we're trying to show here, that endometrium is getting thicker and thicker and thicker under the influence of the estrogen. Now notice at day 14 where they've drawn this line, that's where ovulation occurs. Because the follicle is just ruptured, you'll notice a temporary drop in the estrogen levels because the follicle just exploded. It's not in the condition to keep correcting a lot of estrogen. But shortly after the follicle ruptures, it gets converted to corpus luteum. We secrete some additional estrogen, so you'll notice it goes back up, as well as the majority of the progesterone. Well, the additional estrogen and the progesterone continue to target the endometrial layer of the uterus, make it proliferate more. It actually becomes secretory and very vascular. These are endometrial glands that form here. All this kind of black-looking network here, that's blood vessels. The blood vessels fuse, making big sinuses in there. So the endometrium is very well vascularized. But again, as you move towards the end of the cycle, day 27, 28, estrogen, progesterone levels drop because most of the estrogen and progesterone is coming from the corpus luteum. There's not enough LH to keep it, so it shrivels up and loses its function, stops secreting estrogen and progesterone. Those levels drop. Then, of course, once they drop, menstruation occurs. And you just keep repeating. All right, so go through one more time. We'll kind of go through them both. Up and down a little bit because they're happening concurrently. These times are insane. Both these occur at the same time. Let's look at 2 scenarios. No pregnancy. What would happen if there is a pregnancy? Those things have to occur a little bit. All right, first of all, start at day 1. Menstruation is occurring, so you're swapping off the non-functional layer anymore. And estrogen progesterone levels are low. FSH, LH levels are low. As we go from day 1 on, heading towards day 13 or so, of course, FSH, LH levels go up. That targets the follicle, becomes a primordial. They're secreting a lot of estrogen. So as the estrogen levels go up, that targets the functional layer of the endometrium. It gets thicker and thicker. The cells proliferate more and more. We reach the midpoint of the cycle. You get that surge in FSH and LH. The follicle ruptures. Now this oocyte is going to make its way to the uterine tube, where it's not getting fertilized, so it'll be moving for a few days. It takes like 3 or 4 days to get the main body of the uterus. In the meanwhile, luteinizing hormone targets a ruptured follicle, which causes the formation of the corpus luteum, sometimes called the luteum phase. The corpus luteum secretes some of the estrogen and much of the progesterone, which continues to target the endometrial layer. So for the next few days, it's proliferating more, getting secretory. But then, about 10 days after ovulation, the corpus luteum loses its function because the LH levels are at the lowest point. Corpus luteum then regresses. It stops secreting estrogen and progesterone. It can no longer support the endometrium. Menstruation occurs. So if there's not a pregnancy, they'll just keep repeating about every 28 days. Now, let's see what happens if there is a pregnancy. We'll have to change the pregnancy. All right, first part stays the same. Menstruation is finishing up, FSH LH levels go up, you stimulate the follicle, it begins to grow, it secretes a lot of estrogens, targets the endometrium, gets thicker and thicker and thicker. Now, you reach the midpoint, same thing. The spike in FSH LH causes ovulation, but now this oocyte is going to be encountered by a sperm. So now you got the zygote. Now that's going to take about 4 days before it gets to the main body of the uterus, so not much else will happen the next 4 days. The ruptured follicle, starts to fill into cells and becomes corpus luteum under the influence of LH, more estrogen, progesterone, make things proliferate more and more. But now here's the first potential problem. So now in about 10 days, that endometrium should have made it. That oocyte should have made it to the endometrium. After 4 or 5 days, it should be in the main body of the uterus. So it's now in the endometrium, and this is where the embryo will actually contact the endometrium. And you remember that trophoblast we talked about that hour? That actually grows aggressively into the endometrium. It's often described as growing like an invasive tumor. So yeah, I'm not sure more like parasites. But anyway, you get this projection of cells growing right into the endometrium. It actually destroys the endometrial tissue, so you get all kinds of blood forming out here. That's the beginning of implantation. That's what's going to be the placenta eventually. But here's the problem. Once we get to this point, the corpus luteum, LH levels are dropping, the corpus luteum will regress. If that corpus luteum regresses, loses its function, estrogen and progesterone levels drop, menstruation occurs, out goes the old endometrium and the embryo with it, and there wouldn't be a successful pregnancy ever, and we'd be extinct. So something different has to happen at this point. That's this last one. That wasn't on your hormone list for the endocrine system. This is an abbreviation for human HCG is an abbreviation for human chorionic gonadotropin. This is secreted not by any maternal tissue, by the embryo. When this sucker implants in here, it just starts secreting the hormone HCG, which makes its way to the mother's blood. So I don't know if you technically call this a hormone, but it's a hormone. Not secreted by maternal tissue, it's secreted by this implanted embryo. Now, what does human chorionic gonadotropin do? It is equivalent functionally to LH. Human chorionic gonadotropin has the same impact once it gets into female circulation as luteinizing hormone, which means it will travel in the female's blood to the corpus luteum. Now it doesn't matter if the female's luteinizing hormones drop, because this is basically fulfilling that role. That human chorionic gonadotropin will maintain that corpus luteum for about 6 months. So as long as that HCG is being secreted, it'll keep that corpus luteum alive and functioning for 6 months, which means levels of estrogen and progesterone won't drop because the corpus luteum doesn't degenerate, so there will be no menstruation, which makes for a much more successful pregnancy. So what are we missing now? One more thing missing. How long is our gestation period?
9 months.
We're missing a few. 6. The last few months, the placenta itself, once it develops, which is going to be in here somewhere, that also has an endocrine function. There is placental estrogen and progesterone. Once the placenta develops sufficiently, it secretes its own estrogen and progesterone, which of course maintains the endometrium for the remaining 3 months. So you get estrogen and progesterone eventually from the placenta, then you don't need the HCG anymore or any of this because it's coming directly from the placenta. That is the endocrine control of the female reproductive system. A little more going on in the male. The male's kind of going that way, but those are all the steps. Now, I do like to ask that as a short answer, at least parts of it. So, at the very least, make sure you can pick out, the high points. I mean, where does estrogen come from? Mostly the follicle, some of the corpus luteum. Where do you get most of the progesterone? The corpus luteum. What causes ovulation? It's that surge of FSH and LH at day 14, roughly. What causes menstruation? It's the fall in estrogen and progesterone. So, you can go walk through the whole thing in steps if you want, but at the very least, I'm going to ask you about portions of it. So you're like, what's going on when and why, which hormone supports which part. Also, because of this, if I remember right, some of you might have been aware of this, but really physiologically, males have brain damage. They're born with brain damage. What's controlling the release of FSH and LH? The anterior pituitary, right? Well, what controls the anterior pituitary? Hypothalamus, right? Which is part of the brain. Well, it turns out in an early embryo, there are two regions, two populations of cells, somewhere up here in the hypothalamus. You got a population here, you got a population here. These two populations of cells, they're what stimulate the anterior pituitary, via the portal system and the rest of the hypothalamus to release FSH and LH. One of these areas, one of these populations is the cyclic center, the other one is the tonic center. The tonic cells just allow for sort of constant low-level release of the FSH and LH. The cyclic center, as the name implies, is cycle. It'll dump out a bunch of signal, then none, bunch of signal, then none. So this was the cyclic center. Females have both populations of neurons. These are neurons are in your brain. In a male, one of the things that happens in development, those cells are destroyed. They're brain damage. But of course, they're destroyed because males aren't cyclic. They don't need the cyclical release of FSH and LH like females. So that population of cells, that's part of what happens in males. And this control mechanism is just not there. All right, I think that should do it for the reproductive system. So the last thing we got to do, I have in here, yes. All right, we'll look just briefly, quick run through of development. And of course, call it human development, animal development, or embryology. They're all kind of the same thing. Now, I guess technically the way embryology starts is at conception. When that sperm fertilizes that first egg, you end up with the first cell of a new individual, genetically distinct. That's what we call the zygote. I think you had that on your last. I'm going to step back a little bit because I want to look at fertilization because there's some interesting things going on. Well, basically, development is from the zygote until you have, basically until you're dead, technically. But we're only looking at really early development. We're going to start in the zygote and go through the formation of the first organ system or two, and then we stop. Because honestly, in a human, after 8 weeks, you have all the parts. After 8 weeks, to me, it's what happens? Well, it grows and gets bigger. Then what happens? Well, it grows and gets bigger. Then what happens? It grows and gets bigger. There's some detail, but not much going on. But the early development is, it's lots of different things going on. So that's where we'll start. But we got to step back a little bit and look at the actual fertilization process first, because there's a couple interesting things occurring there. First of all, the reproductive cells. Of course, it's sperm in the male. Here's an electron micrograph of sperm, the oocyte or egg in the female. I think I already mentioned sperm are really, really tiny. These are the smallest cells in the human body. You look at it with our light microscopes, and you can see them moving. You can see them, but that's about it. That's what we use electron micrographs. And we already looked at the parts. The head, basically, you got a big nucleus. That's the genetic material. That's going to be the only contribution of the offspring. There is a little membrane-bound structure up here that will be important here shortly for fertilization. That's called the acrosome. And then you got the midpiece with some mitochondria and the flagellum for movement. Once the sperm are released, they swim through the female reproductive tract and eventually make it to the egg. I think I mentioned, the reason these are really small is during spermatogenesis, the way they're produced, the sperm cells slough off most of the cytoplasm. So they're really, really tiny
It's because they don't have any cytoplasm. Kind of nucleus and the flagellum sticking off and a couple mitochondria. So they are very tiny cells. And also, keep in mind, sperm as well as eggs, they're genetically equivalent. You remember haploid, diploid, one, did you do mitosis, meiosis in one? Okay. Sperm are the only cells the male has that are haploid. They're the only cells produced by meiosis. So rather than 46 chromosomes, of course, sperm only have 23. Oocytes genetically are equivalent. Genetically, they're also haploid. So as far as the chromosomes, sperm and eggs, same contribution, 23 chromosomes each. But when you look at the other contributions, it way favors the oocyte. Let me stick this over here real quick. Now this kind of gives you an idea of the size. They're showing me one oocyte and 500 sperm trying to fertilize it. The oocyte, it's actually the blue shaded area. They are huge. I mean, compared to a sperm, they're gigantic. And even a human egg, well, human eggs aren't particularly big. They're large enough to see. You can describe them as being about the size of a period on a printed page. So you can see them. You saw them on kind of the ovaries. Those are cat oocytes. But they're fairly large cells. Part of the reason they're large cells, again, eggs are produced by meiosis. So you probably went over meiosis, all the nuclear events, right? You get the first division, the second division, you end up with a half. Well, they don't look at oogenesis, the actual formation of the egg. If you think about it, you start off with a parent cell and it undergoes meiosis, meiosis 1. You'll end up producing 2 daughter cells, right? Meiosis 1, you produce 2, then they divide and get 4. And that's true. Genetically, you're going to produce 2 cells that have an equivalent amount of DNA. Those are the 2 daughter cells producing meiosis. However, in oogenesis, meiosis occurs, but that cytokinesis that occurs when you physically split the cells is not equal. The 2 daughter cells, you'll notice, one of them gets all the cytoplasm, the other one doesn't get squat with 23 chromosomes. There's a name for this daughter cell. It is a polar body. While it's genetically equivalent to the bigger daughter cell, it can't do anything. It can't develop. Even if it were fertilized, it's just going to degenerate. It doesn't have any energy growth in it. And of course, then this thing will undergo meiosis too at some point. And it'll do the same thing. One big daughter cell, one little one. And actually, even the polar body may divide in meiosis 2, 1, 2. So you end up, as you would expect from meiosis, producing 1, 2, 3, 4 daughter cells. They are genetically equivalent. However, 3 of them are polar bodies, and they're just going to degenerate. They can't develop, they can't divide anymore, they're done. But that's partly why oocytes are so big. All the cytoplasm that was going to be split between 4 different cells all ends up in one. And that makes them, pretty substantial sized cell. So you see, that's a pretty big cell, and there's a little tiny sperm trying to get to it. So oocytes big. Yeah, genetically equivalent to sperm, but cytoplasmically far superior. In fact, in many species, early development is actually controlled by the cytoplasm in this early egg. You can actually take out the genetic material in some eggs, and they will undergo the first early developmental stages, because that's not the embryo's genes being expressed. It's preformed messenger RNA that the mother stuck in the egg to begin with. So there's a lot of, in many species, what's called maternal dominance. A lot of the early development is actually controlled by the mother. It has nothing to do with the embryo's genetics. Now in humans, we have a little bit more influence. If a human is genetically not viable, development doesn't go very far. You don't have quite as much maternal dominance. The other thing you might notice on this diagram is we're looking at more than just the oocyte here. So let me list a couple of these parts here. The oocyte, this is the plasma membrane right here. So that's the end of the oocyte. You notice there's a couple layers of junk out here. But there's stuff that is extracellular, outside of the egg, not part of the egg or oocyte cell at all. This one, kind of shaded in yellow here, zona pellucida, it's basically some proteins, almost a colloid. It's a protein sort of that incorporates it all liquid into it. They call it the zona pellucida. So this is secreted on the outside of the cell. So it's just a non-cellular proteinaceous layer coating on the outside of the egg. Then this part here in red, you notice this is cellular. These are trying to represent individual cells. You know what that is? What would you call it? We actually named this in lab, I think last time. Remember what that is? The corona radiata. Remember when that ring of granulosa cells around the oocyte, when ovulation occurs, that's released with the oocyte. So this is actually, these are maternal cells. Those are granulosa cells on the outside. Now this diagram is using a different name, cumulus, which is cumulus mass. You have corona radiata, granulosa cells. They do those through formation. But those are maternal cells on the outside. Now the reason this is important, interesting, is in order for fertilization to occur, the sperm's plasma membrane must contact the egg's plasma membrane. The problem with that is you've got these layers of material between it. So the sperm out here have to be able to make their way all the way through these layers of junk or there'll be no fertilization. Now I'm going to use a few diagrams in here. They're not out of your textbook and they're technically not humans. There's just not any good ones in your textbook of early fertilization, like one or two and then a few like that. So I use some embryology in another class. It's more comparative, so we can look it up critters. And I will tell you, early development, all the information, most of the names, the process don't come from humans. As you can imagine, do people do a lot of studies at the universities on human embryos? No, there's medical issues there, it's a hot button topic, no. And honestly, we're placental mammals. The early descriptive embryologists, which is what we're looking at, we're just descriptive, we're not looking at molecular events necessarily here. It's really hard to work on a placental mammal. Why? Because the development all occurs inside, and if you can't take the embryo out, it won't develop. It was technically difficult. So much of the early development was using models that aren't placental mammals. Only when we have the techniques got a little better, we start looking at some mammals, and now we've got little cameras and probes and watch everything internally. But it's difficult to work on placental mammals. So a lot of the early work and early discussions wasn't done on primates, wasn't done on humans. So I say that because I'll use some diagrams like this that have different names on them. Ignore them. They are homologous structures, but when they start working with placental mammals, they kind of give everything a different name, and they aren't exactly the same structures. There's a reason they're different. So I'm going to rename these here just to make them match. This is actually an amphibian embryo or a starfish. I can't remember which. But this layer of green, that is what we would call the zona pellucida. It's called the violet envelope in most of the other embryos you would study. But functionally, it's pretty much the same. It's a layer of, protein kind of around the egg's, plasma membrane and brown. So it's functionally the same as the zona pellucida. They call it something else in the non-placental, but we're going to ignore it. Also out here, they're calling this layer, this one is, it is different in composition. The jelly layer, you've seen frog eggs, right? This is your lily poke with a tongue or something. A lot of other animals, fish, frogs, a lot of other vertebrates, they just put a jelly coat around the outside of the egg. Unlike, placental mammals, that actually is maternal cells out there. The barrier function is similar, but of course the structure is completely different. So you can imagine this is going to function like the cumulus. It is not granulosa cells, but it has the same purpose. So I use this one because this slide shows more stuff. I have the human one. So basically you got this outer layer here, an outer layer here, and then you have the cell membrane. So the sperm basically have to get through those two outer layers, the cumulus and the zona pellucida, before they can contact the egg cell membrane and you get fertilization. Well, this is what the acrosome is for, at least in part. When the sperm swim, of course, they're attracted chemotactically to where the egg is. So they will swim up the female reproductive tract. They will swim up the uterine tube until they reach the egg, which should be, if the timing is right for a successful pregnancy, about the first third of that uterine tube. And when they contact the egg, they will undergo something called an acrosomal reaction. Well, it's kind of cool. If you took fresh sperm and you took a fresh egg and you put it in a petri dish and you dump that fresh sperm into that fresh egg, would it fertilize it in a human? No. It's just kind of weird. Why not? Human sperm, probably all mammalian sperm, but I'm not 100% sure. Right when they're ejaculated, they can't fertilize an egg. They have to call it capacitated before they can fertilize an egg. It turns out the cell membrane, you look at a sperm, you got that acrosome, and of course that's a membrane-bound organelle. There's the head of the sperm. The membrane of the sperm, it's a phospholipid bilayer with cholesterol in it, like you see for the structure of any plasma membrane. However, when sperm are first released, there's a lot of cholesterol in the phospholipid bilayer, in the membrane. If you remember, one of the functions of cholesterol in membranes kind of makes them harder, a little stiffer. And it turns out fresh sperm have a lot of cholesterol in them. And they actually can't do anything if they contact an egg initially. They actually have to swim through the female reproductive tract. That kind of erodes out a lot of the cholesterol. So by the time they reach the egg, they're now considered capacitated, and they can undergo an actosomal reaction. So you either have to chemically treat or allow the sperm to swim through the female reproductive tract. Otherwise, they can't carry out the next step. So we'll assume this sperm is capacitated. It's all ready to go. It's swim through the female reproductive tract. We've eroded some of the cholesterol. As soon as it contacts these outer layers, it will undergo an acrosomal reaction. And all that is, the acrosome, which is a membrane-bound organelle, it contains a bunch of basically hydrolytic enzymes. They call them acrosomal enzymes. But these are enzymes that will chop off organic molecules. So enzymatically break them down into their component parts. So when the acrosomal reaction occurs, kind of what they're showing you here, those acrosomal enzymes, they digest their way through, the outer cumulus cells, the material holding those cells together, the jelly coat in this case, zona pellucida. So the acrosomal enzymes are actually eroding these outer layers of eggs, getting closer and closer to the cell membrane. That's partly why, what would happen now if we took a sperm that's ready to go, it's all capacitated, put an egg in a petri dish and drop that one sperm into the egg, would it fertilize it? Yes, no, maybe? We don't have to do that experiment, but it's unlikely. I guess my question is, why? Females, nice and efficient, ovulate one oocyte typically. How many male gametes are released? 400 million. That's wasteful. Why 400 million? Why not 2?
Because they can't make it.
There's a number of reasons. One, sperm aren't that bright. Some of them just die in the female reproductive tract. Some of them get confused chemically, start trying to fertilize epithelial cells. Some of them go down the wrong uterine tube when they're done. But still, when you get the oocyte, there's still thousands and thousands and thousands of them. Why so many? It's probably the enzymes. Imagine you have one little sperm contacts that egg. That's all that's left. He releases enzymes. Well, sperm are tiny. How many enzymes are in that acrosome? It doesn't be a whole lot. And these enzymes, when they're released, one, they can get enzymatic, they can get neutralized by the proteins in the environment. They can also just diffuse away. So you have one little sperm, it can go, the acrosomal reaction, digest, and then the enzymes just dissipate and neutralize, and then that's it, no fertilization. It seems like a large number of sperm are necessary, certainly more than one, because they all sort of collectively erode through, until finally one of them, so time sequence, but I'll pretend that's a different one, contact the egg cell membrane, and then it will fertilize the egg. And basically all it does, that's why I don't like these models up the left, basically the genetic material is released. The rest of the sperm isn't in the oocyte, it just degenerates. It doesn't contribute anything except 23 chromosomes. Nothing else in the egg or embryo comes from the male, just those 23 chromosomes. But here's also another cool thing. I think this is pretty neat. If you notice, kind of projecting from the egg cell membrane up into these outer layers, there is a particular type of receptor called the binding receptor. Kind of cool process there. Imagine, we'll go back to our petri dish. You take a human egg, put it in there, and then you get a bunch of capacitated sperm all ready to go, hundreds of thousands of them from a hamster, and you dump it in there with that egg. Will those hamster sperm fertilize that human egg? You get hamster board, you can see it at the checkout sign, no. Well, why not? Sperm is sperm. For some reason, well, there's a good reason, you can only fertilize your eggs with basically the same species of sperm, or at least a close, allied species. So sperm have special proteins on their membranes called binding proteins. Not very creative here. The female on the egg cell membrane has binding receptors. So they're very specific, kind of lock and key system. You need to have the binding proteins that are complementary with the binding receptors on that egg, which means you have to be the same species, or at least genetically you have the same receptors and binding proteins. Otherwise, the sperm can't fertilize the egg. Why do you have a mechanism like this? Because you think, especially with, most mammals, we have internal fertilization. You know where the sperm come from that should be getting up in here. So why in the wild is lock and key receptors, proteins? It could be a variety of reasons. One is it prevents you from wasting your reproductive output. Use something else as an example, not a human. Look at a lot of marine critters, sea urchins, worms, corals, clams, oysters. There are male and female of those species. But if you think of an oyster or something, they don't hop around on the beach and copulate somewhere, right? They're all broadcast spawners. They're external fertilization. At a particular time of year, it's usually, based on the tides. In that particular area, all the male clams, let's say, release their sperm. All the female release their eggs. But it's usually not one species. A lot of those marine species key in on tide. So you get a nice big king tide or something. Not only are all the clams and oysters releasing their sperm and eggs, all the coral are releasing their sperm and eggs. Sponges, sperm and eggs. Marine worms, sperm and eggs. It's basically the ocean's like a soup of various species of sperm and egg. All mixing up. Well, if you're a clam, you don't want some worm sperm to fertilize your egg. But you can't prevent it. They're all floating in the ocean. But if that happens, they're not genetically compatible. That egg just won't develop. It's wasted reproductive output. Eggs are kind of expensive in a way, energetically. You put a lot of nutrients in them. You don't want them just out there getting fertilized by some random species sperm. You won't have any reproductive output. That's partly why you have this species recognition. Your eggs are only going to be able to be fertilized by species, by your own species. So in the human sperm, there's a recognition between the binding protein, binding receptors, and that's when that sperm will fertilize the egg and release its 23 chromosomes. What is kind of weird though, and they have done these experiments, you can take a human egg, strip off the zona pellucida, chemically remove it, then you can put hamster sperm with the human egg, and it will fertilize the egg. If you remove those receptors, you can get hamster sperm. No, it's not genetically compatible with that itself. All right, so that one sperm fertilizes the egg, and there we go. That's fertilization, that's the zygote. But there's another potential problem. There's still, 98,000 sperm trying to fertilize that egg. Now, we took 23 chromosomes from the egg. We had another 23 from that first sperm. That's great. We're now back to the diploid chromosome number of 46, right? Now, chromosome number is critical. If you just get one extra chromosome, what is that? One extra chromosome is a 21. That's Down syndrome. Causes some, significant developmental issues. You need to keep the chromosome number at 46. Problem is, there's a bunch, thousands of other sperm trying to get in there. If another sperm gets in there, now you're on 46, you got 50, 69 chromosomes. That embryo won't even develop. So what has to happen here? You have to ensure one sperm fertilizes the egg and no more. So there have to be some things called polyspermy is what they call more than one sperm fertilizing an egg. If you get, one sperm, that's fine. If you get more than one sperm to fertilize an egg, it's polyspermy. That embryo will not develop in most species anyway, certainly not in humans. So somehow you have to be able to block this. Do another diagram. If you look at these PowerPoints, I did post them. You can kind of ignore this one. There are two ways to block sperm from fertilizing the egg. One is called fast block. For years, they thought humans had fast block till they actually measured the egg and then they found we don't. Most species, right when the sperm fertilizes the egg, you get an electrical change across the membrane. It's just a depolarization. It's like you get nerve cells. That's what they're showing you here. That temporary change in electrical potential kind of temporarily delays any sperm from getting in, so it temporarily blocks other sperm. Then they pull human cells out and measure it, fertilization, got nothing. So we don't have fast block. That's why all we have is slow block. Why don't you call it block? Because someone already had fast block. We have something called slow block to polyspermy. That's what this diagram is showing you. Just got a couple of steps. But again, the layers, let me rename them. So the green sort of layer here, this is not the vital envelope. For us, that would be equivalent to the zona. Pellucid, again, this is another frog area. So right about here is the zona pellucid. The jelly coat, of course, it's functional equivalent to the cubulus. So all I have to say, again, I'm using frog embryos as we have. So here's that one sperm, comes in, fertilizes the egg, it's dumped in, it's genetic contribution. Now, a couple things you'll note. Right under the membrane of the egg, you see there's a bunch of circles on this diagram. These are vesicles. They call them cortical vesicles or cortical granules. With the old microscope, vesicles look like dots. You'll see them called granules a lot. It's the same thing. So this is a little membrane-bound packet of material in here. That green junk is whatever they contain. Now, it turns out when that first sperm contacts the egg cell membrane, it doesn't cause an electrical change in humans, but it does cause an influx of calcium ions. Just like in the synapse, calcium ions are common signaling substances. So when that first sperm contacts the egg cell membrane, you get this calcium flux in.
And what that does, kind of like in a synapse, it makes these vesicles migrate towards the egg cell membrane, what is shown here, fuse with it, and release their contents outside of the cell. So these vesicles contain a mixture of enzymes and other substances in these cortical vesicles. But the act of fertilizing from one sperm causes a calcium gets open, calcium floods in, these vesicles migrate to the membrane and release their contents. Now, where they release their contents is important to note. Notice the material from these vesicles is released kind of between the egg cell membrane and the zona pellucida, so it's released in that space. Now, normally, the zona pellucida is held to the egg cell membrane really tightly. In fact, you see these little brown lines are showing right here? Those are proteins, or representing proteins, that extend from the egg's plasma membrane into the zona pellucida. Those two pieces, layers, are normally locked together by proteins. So you really couldn't peel off the zona pellucida or the plasma envelope in this case. It's physically attached to the egg cell membrane. Well, the contents of these vesicles, what it does, it digests or breaks all these proteins. Snap, 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 snap. So it's basically digested the connection between the membrane and the overlying zona pellucida. So they're no longer physically connected. Next, the contents of these vesicles are hypertonic. They're fairly concentrated. So as you secrete that material in here, what they're showing you is there's an influx, an osmotic influx of water. Well, as that water flows in, it expands those two layers. So you notice it's getting really far apart here. It's pretty close here. The contents of those cortical vesicles are hypertonic. It causes the osmotic influx of water, which physically pushes these two membranes apart. The membrane one direction, the overlying zona pellucida, and jellycoat the other direction, or cumulus. And the reason that's important, what's out here? Well, this is where the rest of your sperm are. They're trying to get in, and what you're doing is you're physically pushing all those membranes with all the sperm attached to them further away. And then the last thing that happens, the contents of those cortical vesicles chemically alters the zona pellucida, basically like tans the protein, it locks them together basically permanently. And the zona pellucida is converted into something called a fertilization membrane. The fertilization membrane now is impervious to acrosomal enzymes. So now it doesn't matter how many sperm are out there, they could dump their acrosomal enzymes on this thing all day, it's not sensitive to them, it won't digest the way through. Once that fertilization membrane is formed, it's pretty much permanent. There aren't going to be any other sperm getting in there, even though there may be still tens of thousands around. And what's kind of cool is just got a minute on this. I've got one class I teach every once in a while where we do, what happens to the binding receptors? It looks like do they go away? Yeah, binding receptors run through and are actually incorporated into the zona pellucida, so I assume you're pushed off with it, or maybe incorporated into the fertilization membrane. But what you can do with these things, like I said in another class I teach, we use some in vitro fertilization. It's really easy, of course, we don't use humans. But you can take eggs, they're actually out of sea urchins, you just shoot up the sea urchin with potassium chloride and it spews out its egg or sperm, whatever its sex is, you can't tell how okay. We put them on a slide, you can watch them fertilize, and you can tell when an egg is fertilized. We got this right here. If you look on this slide, you can see, first of all, these little black dots, those are sperm. There's a lot of them here. But these are now fertilized eggs. I have a video to show, but I don't have it handy. Normally, before these are fertilized, see this kind of clear halo around them? That's the zona pellucida, or vital envelope. Normally, it's physically attached to the egg cell membrane, which is here. So if it's an unfertilized egg, you don't see anything. It just looks like this part. But as soon as a sperm fertilizes the egg, you'll see this membrane pop up in one area, and it just kind of goes around the whole egg. You can see it move out. As soon as fertilization occurs, you'll see that membrane gets pushed away from the egg, and then of course it's hard, you can't see that. You can see that whole sort of halo of fertilization envelope form. And what's cool, we use this for a guide in early development. This fertilization membrane is fairly rigid, it doesn't change size. But as we'll see, it also prevents tubal pregnancies in humans. This, while it will prevent any sperm from entering early development, it also prevents the egg from implanting, which it doesn't want to do for about 4 days, so it makes it from the main of uterus. Okay. We'll stop there. So what we've got Thursday, assuming we're here, I should be able to finish development. I don't think I have much more now of development left, because you've got a lecture and a half. That's all I have. So we will definitely have all the test material wrapped up Thursday, assuming we're here. So what I'll do Tuesday, I will review, go over some stuff, give you examples of kind of tedious test questions I put on the exams. And then if I start new stuff, it won't be on the exam. I might start the circulatory system hard a bit in lecture, but that's not for the exam too. That's all. I will see you upstairs in a bit. We will go through the marks.